and I would like to walk you through the usage of cBioPortal and Chromoscope to study the patterns of structural variants here in cancer genomes. And to do that, let's first select a study with whole genome data, the pan-cancer analysis of whole genomes. cBioPortal shows the overview of the cohort that consists of samples of different cancer types. In this cohort, let's search for variants in the BRCA1 gene. This gene is involved in DNA repair, in homologous recombination, and when lost, leads to deficiency in DNA repair. And you can see samples with different types of alterations and mutations. In this gene, homozygous deletions likely lead to loss of function, pseudo frameshift mutations. And we can see two samples with splice site mutations. And we can legitimately question whether this type of alteration leads to loss of function. Let's select one of these samples with a splice site mutation. This is the case view for this patient. It's a breast cancer, 82 years old. The BRCA1 mutation is indeed listed as first one with a high allele fraction. And to confirm the phenotype of homologous recombination deficiency, which we may suspect because of mutation BRCA1, and if this patient was considered for a therapy like PARP inhibitors, we want to confirm the phenotype. And one way to do that is to use cBioPortal's mutational signatures tab. We can see this bar plot, 96 bars, 96 channels, each corresponding to a type and context of a mutation. It's a relatively flat pattern here. And the algorithm, when analyzed this data, concluded that the vast majority of mutations can be attributed to signature 3, which is thought of to arise through homologous recombination deficiency. And that's one piece of evidence suggesting that this sample truly has the phenotype of DNA repair deficiency. Structural variants are as good, if not a better, readout of homologous recombination deficiency. For that, let's turn to Chromoscope. When Chromoscope loads, it shows us the genome view, and that's the circular summary of all chromosomes and all structural variants. The structural variants are visible as lines and circles. And in this sample, there are many green lines. These are the tandem duplications. And it's a very specific type. Their size tends to be within a few thousand of base pairs. These are characteristic of samples with true loss of BRCA1 gene. Let's now inspect the structural variants in more detail. And if we want to inspect structural variants on a single chromosome or region, Chromoscope's variant view is the best way to do that. Here we can see individual structural variants and how they coincide with genes and copy number changes. For example, this structural variant coincides with a large region of loss of heterozygosity. In breast cancers with homologous recombination deficiency, PTEN is a tumor suppressor that is commonly lost. Let's check it in this particular sample in the search box, searching for PTEN. And we do see here one of the typical tandem duplications coinciding with two exons of PTEN, likely leading to loss of function. This concludes a walkthrough of how a mutation in BRCA1 may lead to the phenotype of homologous recombination deficiency, visible to mutational signatures and patterns of structural variants, as captured by Chromoscope. Thank you.